Hello, my dear students. Today we are going to talk about the important topic, that is search for genetic material. There are certain important experiments which were done to prove what is genetic material. So Griffith's experiment, that is transforming principle, is one of the important things. And then DNA proved as a genetic material by Avery, MacLeod, and McCarty. This is also important. Although Mendel explained inheritance and Miescher discovered DNA, and scientists initially believed proteins were genetic materials due to their complexity. By the early 20th century, chromosomes were identified as carriers of genes, but the exact genetic molecule was unknown. Now, when we talk about Griffith's experiment, this was done in 1928. This was the basis for proving that genetic material is DNA. Here, organism that was used was Streptococcus pneumoniae, and S strain was smooth strain. It was virulent strain, and it kills the mice if it affects the mice. Then R strain was rough strain, non-virulent, and does not kill the strain. That means if S strain is affecting the this mice, the mice will die. If R is affecting, then mice will survive. Now, what they did, that they individually injected these things into the mice. When they injected S strain, so mice died. When they injected R strain, mice survived. But the thing is that when they injected a heat-killed S strain and live R strain, they killed this S strain, and then live R strain, they mixed them together. The mice died. And it was told that time that something from the S strain, it got inserted into the R strain, and it transformed this R strain into the S strain. So this is very important. So conclusion, a transforming principle converted R strain into the S strain. So here the chemical nature of genetic material was not identified. What was the genetic material actually? But something was there, that something from this S strain got injected into the or inserted into the R strain, and it actually transformed this R strain into the S strain. Heat is used here, heat. So just remember, we are talking about like heat killed S strain here. So if it's asked in the exam, which of the following experiments used heat? So it is transforming experiment, Griffith's experiment. Then number two was, now it was important to find out what is actually transforming this R strain. Then these three scientists, that is Avery, MacLeod, and McCarty, in 1933 and 44, they purified DNA, RNA, and proteins from the S strain itself. They took the S strain and then they purified these things. Only DNA transformed RS strain into the S strain. Then proteases and RNases, there was no effect when they used proteases to digest the proteins, RNases to digest the RNA. These things got digested, and it was proved that these things were not actually the genetic material. DNAs, when DNAs was used to actually destroy DNA, then transformation stopped here. That means that DNA was the genetic material. Now, final conclusion, DNA is the genetic material. DNA carries heredity information. DNA destroys DNA, hence transformation failed. And it was proved that DNA is the genetic material. Now, this is very important, especially this Griffith's experiment. This is very important. Transforming experiment, transforming principle. I hope you understood it. Next, we'll be talking about some other experiments. Thank you very much for watching these videos.